Well, hello, everybody. My name is Mikis Euripides. I'm a policy and public affairs consultant working with the Coalition over the past 18 months. Today, as part of the Coalition's annual Global Partner Meeting, I'll be facilitating this important session on the latest treatment developments in ovarian cancer over the past year. And to do that, we have some highly informative interviews with leading clinicians in the ovarian cancer space. I'm delighted to welcome Professor Kathleen Moore, who is a gynecologic oncologist with Oklahoma University Health Stevenson Cancer Center. Dr. Moore is board certified in obstetrics and gynecology, as well as gynecologic oncology and hospice and palliative care, and has published more than 200 peer-reviewed publications, as well as serving on the editorial board for 4 academic publications. She has a clinical research interest in drug development and phase 1 trials. Dr. Moore, welcome, and thank you very much for finding the time to talk to our partner organisations and patient advocacy community. Today, we are going to be talking about the MIRASOL study, a phase 3 study that reported earlier this year. Can you please start by telling us the full name of the study and the patient group it's relevant to?
The MIRASOL study the formal name is the phase 3 MIRASOL study, and it's a study of mirvetuximab soravtansine versus investigator's choice chemotherapy in platinum-resistant advanced high-grade epithelial primary peritoneal or fallopian tube cancers with high folate receptor alpha expression.
Can you tell us a little bit more about when you talk about folate receptor alpha expression? What sort of percentage of ovarian cancer patients have this expression?
Well, the expression of folate receptor alpha is super common, actually, in high-grade serous, but about 80 to 90% of high-grade serous ovarian cancer will have some folate receptor alpha on the surface. So it's what we call a tumor-associated antigen, which just basically means that it's a protein on the surface of tumor cells. And it's mostly expressed on tumor cells, but it does have some limited expression on normal cells, which is what makes it a good target for a cancer drug because it's so highly overexpressed on ovarian cancer. In the case of mirvetuximab soravtansine, while we actually do have efficacy even at low levels of folate receptor alpha high, the study was designed just to include those tumors with what we call folate receptor alpha high expression, and that is defined that at least 75% of the tumor cells on the slide stain positive with what we call a 2+ intensity or greater. And so that's the population of tumors that were included on this particular trial.
And was this study building on any other earlier studies?
Oh, yes, years and years of work. Of course, like any phase 3, it is based in extensive phase 1 clinical trial work. So there were, several hundred patients treated prior to this. There was, before MIRASOL, there was a first phase 3 study called Forward 1 that was a similar design. It actually included patients with lower folate receptor alpha expression, but importantly, how the expression was scored. That study actually did not meet its primary endpoint. And so we had to kind of restart the whole program. But we learned a lot from Forward. We learned how to score better, and we learned some other things just about mitigation strategies. So the subsequent trials were better. Probably the most important preceding study or almost concurrent study with MIRASOL is the SORAYA study. SORAYA was or is a single-arm phase 3, which is interesting. We had so much data, we could do a single-arm phase 3 that was mirvetuximab soravtansine in platinum-resistant folate receptor alpha high tumors. Importantly, in that setting, every patient had to have been previously treated with bevacizumab because that was meant to obtain accelerated approval in the United States. And so the patients have to be sort of outside standard treatment options, which includes bevacizumab. So that's why that was done in SORAYA, but it was successful in that it demonstrated a RECIST response rate of 32%, and the benchmark historical reference in this population, again, outside of bevacizumab, is 15%. And the median duration of response was just under 7 months. So again, we're looking for better than on average 5 months. So the SORAYA study really accomplished what we needed it to, and resulted in an accelerated approval for mirvetuximab, so that it could be made available in the United States in November of 2022. The, at that time, ongoing MIRASOL study is the global confirmatory trial for SORAYA, so hopefully will lead to confirmed approval and, more importantly, global access to this medication for patients outside the US.
Fantastic. So building on many, many years of hard work.
Many years, yes.
Yes. So can you tell me, in terms of the actual results of the MIRASOL study, what did it actually tell us? Was there anything particularly groundbreaking about the results? Did it meet a key unmet need? Tell us a little bit about what the results said.
The results of MIRASOL were groundbreaking, and the unmet need, when we talk about ovarian cancer, well, there are many. Screening is the biggest unmet need, and the ability to prevent, that's the highest unmet need. Second highest unmet need is more curative therapies in the front line, so our patients don't get to this point of having tumors that are resistant to platinum. So once tumors are resistant to platinum, that is by definition high unmet need. We need to continue to develop medications that are effective, well tolerated, and cobble together longer progression-free intervals so we can keep, again, quilting together these periods of time that lead to improved overall survival until we can cure. And so that's the high unmet need into which MIRASOL came, and it met all of its endpoints, which is important. So progression-free survival, response rate, and overall survival, which I can take you through. The primary endpoint was progression-free survival. So it was mirvetuximab versus investigator's choice chemotherapy, and this was statistically and clinically significantly positive with a hazard ratio of 0.65. So that translates to a 35% reduction in the hazard of progression or death with use of mirvetuximab as compared to investigator's choice chemotherapy. And so even though this was a secondary, it wasn't analytic pre-planned, but it was a secondary endpoint, I do think that this is one of the most profound findings of the MIRASOL study, because in the history of all ovary cancer, we have never demonstrated with a novel therapy a benefit in overall survival in this setting, in the platinum-resistant setting. This is the first study to accomplish this in a biomarker selected population. So that's quite important and hopefully justifies approvals globally so that patients can start testing outside the United States for folate and eventually get access to this medication.
Great, fantastic. I was just about to ask you about that. So now these results have been published, what normally happens now as a next stage, given that the results are very positive? Tell us what normally happens with a treatment like this.
Once you have mature data that's sort of met all its endpoints and you've gathered sort of all the, a lot of times we present data that's positive very early at meetings just so we can sort of announce it, and then there's other work we have to do. Here, a lot of the data collection is quite mature. So it just, in speaking in general, not just about mirvetuximab and ImmunoGen, once you have a positive study that should, in the eyes of the investigators, lead to a new standard of care, a very large regulatory packet is put together for US authorities as well as European authorities, so the FDA and the EMA. This packets are similar but also different. So it's quite a big undertaking. And so that gets done and they get submitted for regulatory approval, and then the regulatory agencies will start asking questions and asking for more data. And, you know, it's a lot of back and forth with any company, and the regulatory agencies just giving them all the data that they need to make assessments about whether they feel the new medication should be approved in a particular country. And then, of course, it's incredibly complex ex-US. Just because something's approved doesn't necessarily mean it's available, because each country makes their own decisions from there about whether or not they're going to make the drug available. The good thing here in the US, if it's approved, you know, it's available through insurances, sometimes with different copays, but we don't have that same issue as in different parts of Europe. One of the big differences for a long time between US and Europe is that the European agencies really wanted to see OS. And so there are many examples of medications that are available here that are not available ex-US because of the lack of OS, overall survival. And here we have that. So we're very hopeful that when these packages go in, and I can't really speak to timing because that's not quite in public domain, but hopefully
Sure,
a PDUFA date, will be announced, and then the public will know when this is going to be acted on by the regulatory agencies.
Right. Yeah. No. So I fully understand, obviously, with the complexities of that regulatory environment in the US and Europe, for example. When you mentioned potentially some regulatory authorities, perhaps in Europe, might need more information about OS, does that potentially mean that will necessitate another study that looks at slightly different endpoints, or not? Do you think?
Well, this study we met OS, so
Okay.
This study should meet. I hope. I mean, I don't know, but I do hope that this study, because it's PFS, overall response rate, and overall survival superiority, that it will meet the slightly higher bar that the EMA holds for approval of new medications through the European Medicines Agency. In examples where you have only a progression-free survival advantage and not an overall survival advantage, sometimes it's just a function of where that medicine is positioned in the natural history of the disease. So, for example, it's common in ovarian cancer for a frontline therapy, maybe to improve progression-free survival, but not overall survival, because the post-progression survival is so long and so many different things happen, and there's so many different variables that, that study has nothing to do with. And so you may not show overall survival just because you don't control like 7 or 8 or 9 or 10 years of therapy for a patient. But without that overall survival data, sometimes medications don't get approved for use outside of the US. So it's a very high bar. Here, we've demonstrated it. And so I don't think there'll be another study needed in this space. There are other studies being done moving mirvetuximab into earlier lines of therapy. But here I'm hopeful that it will be accepted. Time will tell.
Fantastic. Great. Okay, that's all. That's very useful. Just one other question, more kind of generic question. And obviously, you've mentioned this is an international study. How important do you think that global collaboration is when it comes to trying to ensure success for these types of clinical trials?
Well, global collaboration is imperative for drug development, not just in this setting, but really in every setting. Number one, just from a purely pragmatic standpoint, having these large clinical trials open globally gets access for patients to medications, on the clinical trial early, but it also gets physicians across the globe exposure to new medications with novel side effects or other nuances. So get some experience and comfort and awareness of a medication, which you're well ahead of approvals, which can be quite helpful, rather than just launching sort of into the abyss in a place where they've never tested or they've never had to do the eye exams and trying to figure it out. So from a just practical standpoint, that's helpful. But more importantly, people are just different across the globe, different racial admixtures, different ethnicities, different everything. And sometimes you'll see different efficacy or different toxicity in particular populations that you might not have been expecting, and there may be different dosing considerations. We've seen that with oral medications, probably more commonly than IV, but it's not impossible. So really getting a sense of how a medicine works in more than just people of European descent is quite important in modern era drug development. It's hard to do global studies, but it's necessary.
Fantastic. Thank you very much. I'm delighted to welcome Professor Susanna Banerjee, who is a consultant medical oncologist and research lead for the gynecology unit at the Royal Marsden in London. She is also professor in women's cancers at the Institute of Cancer Research and specializes in ovarian cancer and the systemic treatment of endometrial and cervical cancers. Professor Banerjee, welcome.
Thank you very much. Thanks for this opportunity to speak about low-grade serous ovarian cancer.
Thank you. So we're going to be talking about the RAMP 201 trial, but before we do that, could you just briefly give us an introduction to low-grade ovarian cancer? Low-grade serous ovarian cancer.
Okay, thank you. So low-grade serous ovarian carcinoma is a rare form of ovarian cancer, and we believe it accounts for less than 10% of all ovarian cancers. And the significance of this diagnosis is that we know that one size doesn't fit all with our treatment. And for some patients, chemotherapy can work very well. For example, in patients with high-grade serous ovarian cancer. But for low-grade serous ovarian cancer, we know the biology is different, and unsurprisingly, the outcomes from our conventional treatments to date are different. So if I move on to recurrent low-grade serous ovarian cancer, the response rates to conventional standard of care systemic therapy, so chemotherapy, or hormonal anti-estrogen therapy, the response rates to chemotherapy and anti-estrogen therapy are 0 to 14%. We clearly need to do much better. The response rates to MEK inhibitors, or a class of drug called MEK inhibitors, such as trametinib, are more encouraging in clinical trials, and the response rates range from 15 to 26%. But along with these targeted therapies, it's important to think about toxicities and the impact on quality of life. And for example, with trametinib in the GOG 281 study, the discontinuation rates of treatment due to side effects was over a third, so 36%.
Fantastic, thank you. And so moving on to the RAMP 201 study that looked at patients with recurrent low-grade serous ovarian cancer and reported earlier this year. Can you talk to us about the study, your role in the study, and also what the results tell us?
Sure. So going on from what I've just said about low-grade serous ovarian cancer, I want to point out that to date, there are no FDA or EMA approved treatments specifically for this form of ovarian cancer. So this really highlights the unmet need to improve outcomes for women with this condition. And that's what really led to our research efforts to improve response rates and efficacy of new drugs in the clinic through clinical trials for this disease. So RAMP 201, also known as NGOT-OV60 GOG 3052, is a phase 2 study of a targeted non-chemotherapy drugs called avetometinib, or VS6766, with or without defactinib in recurrent low-grade serous ovarian cancer. So this study is sponsored by Verastem, the pharmaceutical company responsible for these drugs, and it really reflects a collaboration across Europe and US, across the globe, really, specifically for women with low-grade serous ovarian cancer. So just to say that this study was designed based on the activity from another trial that your audience may have heard of called the FRAME study, and that was the phase 1 trial that looked at the combination and the doses for combination. And that was a study, an academic sponsored study from our institution, the Institute of Cancer Research. And based on that activity, this led to an FDA breakthrough designation status, and the community of investigators globally to press on with RAMP 201. So this trial that I presented the early results, and that was the initial efficacy results from 64 women in Part A, and the safety data of over 150 patients enrolled. And our data cutoff was April the 6th in 2023. And I presented this at ASCO in Chicago earlier this year. and more recently at ESGO in Istanbul. So the key points are that for this combination arm of avatimetinib and defactinib, the overall response rate was 45% for the overall population, 60% response rate in the KRAS mutated group of patients with tumours that have KRAS mutations, and 29% overall response rate in the KRAS wild-type patient groups, respectively. So the other important point is that responses to avatimetinib and defactinib were observed in 3 out of 4 patients previously treated with a MEK inhibitor. So we also looked at monotherapy with this RAF/MEK inhibitor, avatimetinib, and the response rate there was 10%. So we concluded that the combination is superior and should be the go-forward regimen for developing this regimen going forward in low-grade serous ovarian cancer, and the regimen to go forward with in the phase 3. Now, whenever we think of efficacy, we need to look at safety hand in hand. So the safety profile was consistent with what we previously reported of these drugs in the phase 1. And the majority of treatment-related adverse events were what we call grade 1 or 2, and they included nausea, diarrhea, rash, and fatigue. The most common grade 3 or more toxicities was a change in a blood test called CPK. But what I'd like to point out is that the patients weren't symptomatic of that. And another important point when we think about dose reductions and discontinuations is that for the combination arm, 17% of patients had dose reductions. And to date, at the time I presented this, it was around 12% discontinued due to a treatment emergent adverse event. So finally, what I want to say is a little bit about the mechanism of these drugs. Avatimetinib is a dual RAF/MEK inhibitor. It's an oral agent that attacks this MAP kinase pathway that we know and recognize is activated in low-grade serous ovarian cancer. And defactinib is a FAK inhibitor, which from our studies in the laboratories have suggested that dual combination can overcome some of the resistance mechanisms that we understand may limit single agent RAF or MEK inhibition. So that in a nutshell is a summary of the study.
Thank you so much, Dr. Banerjee. So that was a phase 2 study. What are the next steps, and what do you see as a reasonable timeline before we think kind of patients can make full use of this as a treatment option?
So RAMP 201 is still recruiting. We're looking at various options of doses and such. But this initial results I think are really encouraging. We're really delighted that it worked so well for some women in the trial. And we were hoping that based on the response rate seen in RAMP 201 and also taken together with the phase 1 trial I mentioned, the FRAME trial from the Royal Marsden Institute of Cancer Research, we were hoping that in this rare form of ovarian cancer with limited effective treatment options, that full regulatory approval would be considered or possible. As I said, the combination has FDA breakthrough designation status in the US, so we need to watch this space to see if, based on these results, patients may have the opportunity to access in the US whilst waiting for recruitment and the results of the phase 3 trial, RAMP 301. So RAMP 301, let me mention a bit about that. That's about to open. It's a randomized trial against standard of care treatment, which is chemotherapy or hormonal therapy that I mentioned at the beginning. I personally hope that once we have more long-term follow-up of patients from the existing trials, FRAME and RAMP 201, with the support of patient groups, some countries may consider assessment for approval. But as I said, in the meantime, patients will have the opportunity to enroll in RAMP 301, and this will open in more countries around the world. It's basically hoping to have 270 patients And the important point for patients, in fact, knowing that obviously the activity I've described, is that patients randomized to standard of care existing chemotherapy or hormonal therapy have the opportunity to cross over at the time of progression to ivosidenib with defactinib. So I'm very pleased to be the ENGOT, that's the European Network of Gynecological Trials, to be the lead for this, and I'm very excited about the trial opening and having more opportunities for women with low-grade serous ovarian cancer.
And you've touched on the international global nature of these studies. How important is global collaboration when it comes to addressing unmet needs for patients in this particular space?
It's essential. So in particular with rare forms of cancer. And so we've described that in low-grade serous ovarian cancer. So if we are going to improve treatment options, we need enrollment into clinical trials. And it's not possible to do that from one country or a few countries in a rare condition. Cancer has no boundaries. It's a global disease, and therefore patients being able to be enrolled in individual countries is really important. Also thinking about distance to travel, and I appreciate the complexities of trial sites opening in rare conditions, but I know too well that many patients will travel across an individual country to access entry into clinical trials. So, you know, it's essential. And also we need to learn, or have the opportunity to learn, if there are differences between patients in different countries, different ethnicities, for example, in terms of the amount of benefit patients derive from individual treatments. So that's across clinical trials, I would say.
And just on that point of global collaboration, do you feel it is genuinely global, or is it perhaps still very much a case of higher income, middle income countries that dominate the space when it comes to patients being involved in these clinical trials? Do you think there needs to be something done about that in some way, if that is the case?
So I'm very pleased to be the co-chair of the GCIG, that's the Gynae Cancer Intergroup trials in rare cancer specifically. And I've been part of the group attending this meeting for a number of years, and I can see that it truly is a global effort. You know, there certainly is recognition of where trial enrollment has been traditionally, and the need to expand beyond this, and this is happening. We saw this, I know a different condition, but we saw this in trials of cervical cancer and endometrial cancer reported this year at ESMO 2023, that there is increased global recruitment for these conditions beyond the traditional countries like the US and certain European countries. So it's important to retain that but increase recruitment, and that is in the hands, of course, of our sponsors, which, you know, for example, this trial is a pharmaceutical company, but there's recognition across the board of the need to open across countries. And hand in hand, there needs to be the infrastructure to recruit to clinical trials and support patients so that the data is useful and safety for our patients across opening new centres within existing countries, but also new countries. And I think that's really important.
Finally, one more question. The World Ovarian Cancer Coalition is a patient advocacy group. We have many partners and other patient advocacy groups based in countries across the world. What is it you think that patient advocacy groups can do when it comes to supporting more research to focus on the rarer forms of ovarian cancer, such as low-grade ovarian cancer?
So the patient advocacy groups are again essential and have grown so positively and in the last few years. And there's also the recognition of the importance of patient advocacy groups by regulatory bodies, by funding bodies to access the drugs, and also funding bodies and pharmaceutical companies for making decisions about whether a trial is important for patients, because ultimately we do all this for patients. So we need to know what patients need, what would they like, and how can we then support the patients best. So I think there's a role of patient advocacy throughout the pathway, from the development of a trial through to the information and delivery to patients who are interested in the trials, to be more aware of trials, through to reporting the results and getting the message out there like we're doing today, and then reaching out to patients groups so that patients are aware of trials in rare cancers, so that we're not just reliant on doctors referring to clinical trial centres. And then at the other end, it's about then the importance of patient awareness and lobbying for funding and approvals of these drugs as standard of care. So essential throughout the pathway.
Thank you so much for your time.
You.
Have a good week. Today we are going to be talking about improving ovarian cancer outcomes with artificial intelligence. I'll be referring to that as AI, and specifically about an AI-based model developed by researchers, including our next speaker. Professor Ramona Witek is a professor of radiology and leads the Center for Medical Image Analysis and Artificial Intelligence at the Danube Private University in Krems, Austria. Professor Witek is also Honorary Clinical and Research Fellow at the Cancer Research UK Cambridge Institute, Department of Radiology, and her research focuses on the development of AI-based segmentation tools, radiomics-based predictors in breast cancer and ovarian cancer. Professor Witek, welcome.
Thank you. Thank you very much for having me.
Can you please tell us a little bit about the basics of this specific research that was recently published in Nature Communications, its aim, how it works, and which group of ovarian cancer patients the research targets?
So the work that we did and that we published recently on ovarian cancer was done in collaboration and also during my time at the University of Cambridge. It was a strongly interdisciplinary work, and our goal here was to develop a tool that helps us predict in patients who are newly diagnosed with ovarian cancer and who are candidates for neoadjuvant chemotherapy. In those patients, we want to predict what their chances are of actually successfully shrinking the tumor with this neoadjuvant chemotherapy. And we thought, what types of information do we have about patients at this time point, very early during their patient journey through ovarian cancer? And of course, there's a multitude of data. We have patient demographics. We know who our patients are. We have some information about the treatment they will get, and they all may have had already. Blood samples are usually taken from these patients, and there are more standard tests that can be done on these blood tests. But there are also more advanced types of analysis, like, for example, circulating tumor DNA can be extracted from blood samples. We know that all cancers, or most cancers, shed a little bit of DNA into the bloodstream, and by taking blood samples, we can quantify this DNA that comes from the tumor and is found in the circulation. So these are things that we took into account. And as a standard procedure, patients with suspected or diagnosed ovarian cancer undergo computed tomography or CT scans. usually of the abdomen and often also of the chest. So we also have imaging data for these patients. And there is a relatively new field of image analysis, and that field is called radiomics. And it simply means that we do not only look at the images like as we as radiologists usually do when we report on a set of images. We look at them, we measure the size of tumors, we describe the anatomical locations, and we compare with previous images if they are available. But we don't have any way of describing the texture or the more detailed appearance of tumors. But we know now that there is a wide variety of texture, heterogeneity of the appearance of tumors. And radiomics is a field where we use computers to quantify this texture and the appearance, the patterns that we see within these tumors. So together with the features or the types of information that I described, clinical data, blood-based data, we also calculated so-called radiomics features for all the tumors that were depicted on the CT scans. And then we used artificial intelligence to develop a so-called predictor. What does that mean? It means that we use computer programs to find out which of this large number of features are most relevant to understand which patient is going to have a good response to neoadjuvant chemotherapy, meaning that the cancer is going to shrink, and which of those features or values are less important. And for doing something like this, it's usually advised to have two completely independent data sets. So you use one of those data sets for developing this machine learning tool, and then you have a completely separate data set that you use for testing this machine learning tool, the so-called predictor that you developed, and that is what we did. So from two centers in the UK, we had we included patients with all these data that I mentioned, and our machine learning framework is what we call it. We were successful in developing such a framework, and we could successfully predict the shrinkage of tumor deposits, either measured as a change in the volume or measured as a change in the diameters of disease. And interestingly, so of course we wanted to understand better which type of data is most important. Are the clinical data, the demographic data, the standard blood tests that is done in patients with suspected ovarian cancer, are they enough to achieve good accuracy, a good quality of predictions? And we found out that it's actually the addition of those radiomics features, those quantitative imaging features, that made a huge difference, and that improved the predictions a lot. Of course, circulating tumor DNA is a relatively new approach as well, and we're also very interested in seeing if that could help us as well. Unfortunately, the addition of that type of data did not improve the predictions any further, but it is still possible that assessing circulating tumor DNA during treatment at later stages might be highly informative. But at this very early time point, at the time of diagnosis, for our purpose, it did not make a big difference. But this quantification of the appearance, the patterns that we see, the texture that we see within tumors, that was very important and helped us improve these predictions a lot.
And in terms of, you mentioned centres in the UK, was there also an international element to this research? Were there other countries potentially involved, or are there other countries where this potentially will be rolled out to?
In terms of the patient cohorts, unfortunately there was no international element to it so far. In terms of the research team, it was very international, and several of the researchers involved in this study have moved on in the meantime and have moved to different centers in Europe. And this is what we are planning now. We are planning to continue this work and to improve it, and to use this predictor and to improve it on patients in different countries now in Europe. But we are, of course, very open to collaborating with centers anywhere around the world, because it's extremely important to include a group of patients as heterogeneous as possible, to make all these AI tools proof for and safe for real world applications. Absolutely.
And just more broadly, I suppose, do you think that kind of models of this nature have the potential to be real game changers when it comes to predicting outcomes for patients with ovarian cancer, and how widely do you think this will become available? I suppose perhaps if the research has been done in Europe, that's where this will begin, if you like. But how I suppose, so the first question really is, what's the capacity for this to be a real game changer, and how widely do you think this will become available?
So the clinical use for this could be to gain more information when it comes to deciding whether or not a patient with ovarian cancer should undergo neoadjuvant chemotherapy, which means chemotherapy first and surgery only second, or if a patient should have surgery first and should then get chemotherapy afterwards. So this would be the possible clinical application, and we don't have many tools that do help us with this decision. Since the data we used for the predictor that works best are all data that are acquired in a standard of care setting, I think the potential is huge, because we don't need to do any very expensive lab tests. We don't have to do any extra scans. It's all based on data that are available already in most centers across the globe. So these are really good conditions for applying this at a larger scale. However, our study is a research study, so we need to show that it works in more patient cohorts. And ideally, we use this predictor alongside possibly a clinical trial where we do not make any decisions based on the predictor, but we let this machine learning tool, we let this predictor run alongside a trial, and we see prospectively, so looking as patients come and as patients are included in the study, we check if our predictor makes the right prediction. So that would be the next step, and then we can, if that proves to be successful, then we can start the first clinical trials where we actually base treatment decisions on such a predictor. And in terms of possible difficulties with the implementation of such a thing. For this project, it was necessary to manually outline all the disease deposits throughout the whole body in all these patients. And that is very, very time-consuming work, and it also requires radiologists who are specifically trained for this type of imaging and disease. And we are also working on automating this outlining, and we call it segmentation. So we have published the first work on that as well. So the faster we can make this automated outlining, the more likely it is that predictors like this can be used in not only in research centers and research hospitals, but also in hospitals that are more focused on clinical work.
Thank you, Professor, for taking us through that highly informative research.
Thank you very much for having me, for inviting me, and I'm very happy to do something similar again.
Thank you very much. Take care.
You too. Bye.
Bye-bye.